Hi, I think we're at 12:03, so we should probably get started. So it's a great pleasure to invite Evan Newell to come spend the day here and give us a talk on what he's doing, but also meet with all the faculty. So I'm supposed to give it a little bit of a summary of Evan's travel and training history. So he is actually not Canadian, although he did both his undergrad and PhD in Canada at McGill and then Toronto, and then decided to move to the West Coast and did a postdoc at Stanford with Mark Davis, sort of at the uptick of CIIT, and then made a big change, and for the next 5 years moved to Singapore, where he's a principal investigator at their A-STAR program, and then eventually was just before the pandemic, made the decision to move back to the US and has been at the Fred Hutch since then, and has done a lot of very interesting work on what he's going to talk about is T cells in cancer, but we're collaborating on autoimmune kidney disease now, and so it's great to finally get him here.
All right.
Yeah.
All right. Thanks, Sean. That was very nice, and thanks so much for having me. I had a lot of possibilities of things I wanted to talk about, especially some of the collaborations I'm doing with folks here. But I decided to try to keep things somewhat simple and kind of focus on one of the main threads in the lab, which is to try to profile tumor-specific T cells. And I'll sort of work my way through the ways we're trying to do this. And yeah, it's been great to be in Seattle, just down the street for the last 5 or 6 years. So hopefully I can convince you to work together with me on some projects as well. So I'll just tell you about my general goals for this topic of sort of the cancer immunology work that we're doing. And then I'll tell you about sort of the approaches that we've taken historically to do antigen-specific T cell profiling, and then how this relates to my role in helping to direct the Fred Hutch Innovation Lab, which hopefully you can take advantage of. We're trying to look for collaborations to help, help adoption of new technologies and things. So I'll talk about that, and then I'll talk more about what I'm here for, which is like, how can we effectively study tumor-specific T cell responses, also in the blood, first in the tumor and then in the blood. And then I'll just do a super brief overview of some of the projects and other interests that we have, if I have time, just to let you know that the other types of things that we're doing, because it'd be great to work together as well. So in terms of cancer, I think, given that immunotherapies have so much promise, both cellular therapies and checkpoint blockade immunotherapies, there's a real opportunity to ask questions related to mechanisms of immunotherapy response versus resistance. And there's a lot of possible reasons for why sometimes immunotherapies work versus don't. And I think what we're trying to understand is the role of the endogenous tumor-specific T cell response. And so we want to try to study what are the magnitude, where those T cells can go, and the extent to which T cell responses are dysfunctional in different contexts. And so in order to do this, we want to try to understand what the status of the tumor-specific T cell response is. And I think it's not as easy as you would think, because it's not necessarily simple to identify those tumor-specific cells. So we're trying to find better ways of, identifying tumor-specific cells, both in the tumor and in the blood, and then try to learn from them. And so one of the strategies we've taken, and this will be kind of the last part, is focusing on virus-specific T cells in the context of immunotherapy for virally driven cancer. So we've been working on both nasopharyngeal carcinoma, which is an EBV-driven head and neck cancer, and then Merkel cell carcinoma, which is Merkel cell polyomavirus-driven cancer. And I'm sure a lot of you are aware of work done in Seattle on this with Paul Neum's lab and others. So we've been working with them as well to track these T cell responses. So the vision of my lab when I got started, and also when I was a postdoc, was to try to decode T cell responses in health and disease by sort of first understanding what the range of phenotypes are, and understanding what sort of underlying diversity of T cells, and then how that relates to T cell specificity. So we've been trying to, as broadly as possible, probe T cell specificities as well as T cell phenotypes. And so on the left here is a UMAP plot. I'm going to show a bunch of these. This is something that we really love, even though there's some controversy about this. But basically, UMAP plots are these dimensionality reduction plots that take high dimensional data. And in this case, these are mass cytometry data. So we've measured about 40 different markers on each cell. And then we're taking that 40-dimensional perspective and boiling it down to 2 dimensions. And on these plots, each dot is a cell. And if a cell is nearby to another cell, it means that it's similar in high-dimensional space. And this UMAP is supposed to try to maintain sort of cells that are similar to be very close by. So that's sort of local similarity. And also, if they're very different, they should also be far apart. And what's cool about these plots is that you can see this is analysis that we've done on a bunch of different tissues, and you can see that these phenotypes of CD4 cells and CD8 cells kind of make sense with respect to what we think about the cells are doing. And so like these are naive CD4 T cells in this blob, and then you can see this as they kind of differentiate into many different phenotypes, there's a lot of diversity. And so so anyway, trying to understand what all this diversity means and what all these different populations are doing is something that we've been fascinated by and wanting to then understand in any disease context. If we can identify the cells that we know are responding or specific, and then we can understand what they're doing by figuring out where they are on these maps. And so the way that we've been doing this for a long time is to use peptide MHC tetramers. And so when I was a postdoc, I developed a method to multiplex peptide MHC tetramer staining. So we use, and we're using mass cytometry. You can use flow cytometry, but it works especially well with mass cytometry because what we do is we label tetramers with unique combinations of heavy metals and mix all these tetramers together. And then when we identify a cell that stains with the right combination of heavy metals, we can know what peptide they're specific for or what peptide MHC they're specific for. And so within a single sample, we can look at up to hundreds of different T-cell antigen specificities. And then we have 20 or 30 channels remaining to profile the phenotypes. So we can generate these UMAP plots and then also identify the antigen-specific cells that we can overlay onto these UMAP plots. And so to me, it's exciting to take somebody's blood sample and then look and see really what's happening to their T-cell response against a bunch of different things. So this is just a random example, and actually I think there's some issues with this, but this is a CD8 T-cell UMAP where we can sort of delineate the major subsets of CD8 T-cells. And then the examples on the bottom here are each red dot is an antigen-specific cell for these different epitopes. And so for each epitope, you can try to figure out, what is that cell doing and how does that relate to the status of that infection or anything. And so, again, related to what I've been wanting to do is to take blood samples and screen across all kinds of different things and have a better understanding of what that person's immune system is doing in response to all these different antigens, and then to apply that to different contexts. And so before I get into using that, I'm going to talk about how we're trying to also improve this with other methods. And so about a year and a half ago, I became the scientific director of this Fred Hutch Innovation Lab. And we're trying to help adoption of new single-cell sequencing and spatial transcriptomics methods through collaboration with different researchers in the institute or across the consortium, really. And the goal is to try out new things and sort of de-risk it for labs as we collaborate, and then move those methods over to shared resources to make them more available. And so, for instance, we've done a lot of benchmarking of different single-cell sequencing methods to try to see, oh, there's a new method. There's parse, there's scale, there's BD, there's 10x. Which one works better for the different approaches that people are taking? And can we quantitatively compare these things? So we've been doing a lot of benchmarking, and just recently, there's a lot of new single-cell sequencing methods that we've been comparing. And then the other one that I think is especially tricky to figure out is this emergence of new spatial transcriptomics methods. So we've been trying to do some benchmarking of different spatial transcriptomics platforms and also help people just get going with applying these to their experiments. And I'll show how we've been trying to use this, and there's some example data there. I'll be quick on this part. I mean, we're excited about new technologies for FFPE samples. So one thing that we've helped to demonstrate the utility of was this 10x flex protocol, which allows you to fix the cells prior to doing single cell sequencing, and it's probe-based, and it has a lot of really good advantages. And so you can even work with FFPE samples and get single nuclei and get really good resolution single cell data. So we've been trying to share our experience with that. We've also done a lot of Visium, and this is rapidly evolving, but another way to do spatial transcriptomics on fixed tissue, so FFPE, with the same kind of chemistry. But then the thing, of course, that everybody's very excited about are these in situ hybridization-based spatial transcriptomics methods, and we've been focusing on 10x xenium. We also have a CosMx at the center, so we've been doing some comparisons between those. But I'm going to tell you about our experience so far in sort of applying these and what we've been doing with xenium and sort of some of the analysis methods that we're using, and sort of try to fit that in with the goal of my talk as well. So we've been working on kidney tumors because we were able to get these tumors both fresh and fixed. And so we have some nice example patient samples where we have very good quality single cell sequencing data from fresh samples that are dissociated. And then we've also run all these other platforms as kind of to compare them against each other. We also have done some CosMx on the same samples. And so we're able to get really good single cell data, mostly focusing on T cells in our case, this 10x flex, Visium, and also xenium data. And the purpose for this then is to try to understand, what's happening with antigen-specific T cell responses. And so I'll go back to how we try to profile antigen-specific T cell responses before I'll come back again to these approaches. And so in the tumors, this is pretty old work now, but what we were trying to do, again, now with mass cytometry again, is to use our multiplex tetramer staining approach to identify tumor-specific cells. And so in collaboration with some genomics people, we took the kind of brute force approach of getting a bunch of lung tumors and colon tumors for which there was full exome sequencing. And then we worked with computational people to identify the mutations in those tumors, predict these neoantigens. So we'd look at all the peptides that can bind to MHC class I, and then we made over like 1,000 different peptides for all these neoantigen candidate epitopes. And then for each patient, we'd screen however many there were. There was either like somewhere between 10 and 100 or even a couple hundred per patient. So this is kind of heroic work. And at the same time, we included all these control epitopes for different viruses. So in our screening, we can look at lots of different epitopes. And the challenging thing was, even though we had examples like this is with this combinatorial tetramer staining, and these red dots we were super excited about. This is a really amazing example of a very robust, so we take the tumor, these are ex vivo T cells, so they haven't been expanded or anything. We can identify these neoantigen-specific T cells with our coding, and in this patient, really high frequencies of a particular neoepitope. But in fact, this was a rare event across all the patients that we looked at, and we only have a couple examples where we could actually find these bona fide neoantigen-specific T cell populations. So this is pretty frustrating. But what was interesting is that in parallel to this, what we noticed is that we had a lot more examples of T cells that were specific for these control virus epitopes like EBV, CMV, and flu and stuff. And so Yannick, the postdoc, was actually quite interested in this. And we went on to show that these are very common, that you can have these cancer-unrelated antigen-specific T cells in tumors. And I won't go into the details, but in fact, the phenotypes are distinct for those cells as compared to the tumor-specific ones. And we had shown that they were uniformly negative for CD39. So then, based on this work, we had hypothesized that CD39 would be a good marker for tumor reactivity. And in the meantime, there's been a lot of other papers consistent with this notion. So in fact, if you look at the specificities of T cells infiltrating solid tumors, a lot of times most of those cells are not specific for the tumor and instead specific for all kinds of random stuff. These are just bystander T cells that are infiltrating. And then the expression of CD39 is sort of a marker of terminal T cell exhaustion, has proven to be a really good way to actually identify tumor-specific cells in tumors, not perfectly. And then there's other markers that have since gone on to be more accurate, or in combination with CD39 provide more accuracy. But nonetheless, what's interesting about solid tumors is that, in fact, you've got to be careful when you're trying to understand what's going on in there. There's all these heterogeneous T cells, and in some cases, only some of them are going to be tumor-specific. This is a very cool paper also where they showed that, like us, they could see sometimes very high frequencies of unrelated antigen-specific cells across all kinds of different tumors. And so to me, I think this actually is when I showed some of this earlier on, people thought that bystanders is, doesn't make sense. Why would you have so many bystanders in tumors? But I think to me, it makes a lot of sense. The tumor is an inflamed tissue. And then when T cells go into that inflamed tissue, there's no antigen specificity involved in that, crossing the endothelium into the tumor tissue. And then once they get in there, there's all kinds of inflammatory mediators that can cause them to take on very diverse phenotypes. But only the cells that are actually tumor specific will then take on this terminal exhaustion phenotype. And so we've done more on this topic and shown that CD39 can also be a relevant marker for discriminating bystander CD4 T cells as well. Among non-Treg CD4 T cells, there can be a lot of bystander cells. And also even for mate cells, we showed that these mate cells can be recognizing what seem like bacterial-derived antigens in tumors like colon tumors that have a lot of bacteria. The mate cells are also taking on this more terminally exhausted phenotype. And so with the access we've had to kidney tumors, we've done the same sort of thing. And so in kidney tumors, we can similarly see that there's pretty distinct populations of CD8 T cells that either express CD39 along with other markers of terminal exhaustion, or that look like bystanders and that are enriched for these antigens, unrelated cancer antigen-specific cells. So down at the bottom here is an example of a tumor where we could detect CMV-specific cells, and you can see they take on all kinds of diverse phenotypes, which are different than what you see in the blood. They look like resident cells. They sometimes express CD103, PD1, all kinds of things, but they rarely cross over into this CD39 compartment. And same here for some EBV-specific cells. And so what we've been wanting to do then is, understand, well, what are all these cells doing? And so this would come back to what are they doing in space as well. When it comes to, these are antibody markers, but when we use single cell sequencing on these tumors, and this is what I was talking about earlier, we can identify the same general populations, but then define them also with transcripts. And in fact, this CXCL13 has also become a more accurate marker even for tumor-specific cells. So these terminally exhausted cells, in terms of transcriptomic profiles, CXCL13 is very easy to detect as an abundant mRNA that's expressed only in these more terminally exhausted cells that are also, positive for CD39 and all kinds of other things. And so in looking in the space, we wanted to then just try to understand where these different T cell populations were. And so that's when I'm coming back to, to the xenium data. So for some of these same samples that we have single cell data, we know what to expect in terms of the T cell compositions. We got FFPE samples from the same tumors. And then with our new Xenium, which is about a year ago, we run these samples, and we also included a custom gene panel that had the markers that we knew were important for distinguishing T cells, including CXCL13. And so then the goal was to see if we could sort of parse out T cells in these samples. But then the amazing thing was that these data were actually obviously very interesting on their own. And so I'm just going to talk about sort of our experience in working with these data. So this is a UMAP plot that comes out of the Xenium system. So this is using, and I'll talk about cell segmentation later, but this is using sort of the out-of-the-box cell segmentation for Xenium data. So this is one of those tumors. And what's amazing is that you can profile a lot of cells. So here we have 130,000 cells, and we're only profiling 380 genes. This has already improved quite a bit. And you can make out populations that make sense. We can see different T cell populations, but they're obviously a lot more mixed together with each other than what you see for actual single cell data from the same tumor, but the composition are actually similar. So we're reassured that we're kind of seeing the same populations, but the Xenium data, obviously, because it's sparse and the segmentation is going to be an issue, we can only make out the population so well. And I think this is when we started getting this staff scientist in my lab, Daniel Jones, to work on these data. And he pointed out to us that the way that the 10x Genomics is doing cell segmentation, and they are improving this, they knew that this was an issue, was very naive. So basically all they are doing is identifying nuclei from DAPI staining, and then they just expand out a certain radius until it comes in contact to another cell, and that's it. And then any transcripts that are in that boundaries, they call for that cell, and if it's out, and so it's a very, very naive cell segmentation. Nonetheless, you can see these cell populations amazingly well. So Daniel saw this as a really good opportunity to try to improve things. And he found this algorithm online, and this had been published. I should have the reference, but I don't. Sorry, you can find it. But this Bayes algorithm was amazing. So we put this in, and so Bayes algorithm is kind of a statistical model, and it is able to identify cells by, setting the boundaries in order. So basically, what it does is it's setting the boundaries to find cells that have kind of uniform transcriptomic profiles. So it's a little bit of a circular logic, but it finds regions of space that have uniform transcripts. And then obviously it does really, really well then in sort of in segmenting out cell populations. That makes sense because it finds areas that are uniform in their transcripts. And it actually has quite a lot of assumptions that go into that Daniel was very concerned about. But then the other thing that's problematic about Bayes is that it finds way too many cells. So it'll find little pieces of space that have uniform transcripts, but it can be tiny. And so what he calls it is like, it does a lot of gerrymandering. So it finds regions of space that fit what it's looking for, and it'll call that a cell. And so the cell numbers are like inflated. So they have twice as many cells as you should have. And on top of that, it's extremely slow. So it takes 24 hours to do one run. So this is what he was talking about. It's super slow, which makes it almost infeasible. You can post this, actually, I don't care. And then it finds these tiny cells with almost no counts in it. And so he had interestingly he'd been working with this thing called cellular POTS modeling, which is it's a kind of it's a pretty unique field where they're taking trying to simulate cells. And he was doing this because he had another paper where he was trying to create benchmark data for differential gene expression analysis on tissues, and he was tired of just using like shapes that were squares or circles or whatever. He wanted some cell-looking shapes. And this POTS modeling, you can put in equations to model what cells should look like, and it can quickly generate cell shapes. And so in working a lot and trying to understand this whole cell POTS modeling literature, he decided that he could kind of reverse the cellular POTS modeling. And instead of modeling cell shapes, he could model the sort of same thing that Baser was trying to do, and that looking for cell shapes that fit sort of a uniform distribution of transcript profiles. And what was amazing also is that there's so many people using these models that there's really computationally efficient ways of doing this. So you can run this thing in 20 times faster than Baser, and he can set the sort of rules in terms of how the modeling finds cells to make more sense, and he prevents this gerrymandering. So he gets the right number of cells, it's fast, and then the UMAPs are still looking really good and actually make even more sense than the Baser data that comes out. And so this is some of the data that we'd first generated across 4 different RCC patient samples where we had healthy tissue as well as the tumor. And just across all those samples, merge everything into one big UMAP. And I was really excited about this because you can see each tumor is very distinct because tumors are like that. They're always different. But then the non-tumor cells, you could make out all different populations across all these different samples. And then they make a lot of sense. And then you can take those cell populations that he defined, using the clustering of the, like what was shown in the UMAP, and then go and look at where these cells are. And this is a pretty cool image. We're using this breast cancer panel with some T-cell focused add-on genes, but still we can define lots of different cell types in the kidney, and this is the normal kidney where we can see different populations. You can make out glomeruli and stuff, and this is where Sean can tell me a lot more that I don't understand about the kidney. But for us, it was cool then to look at zooming in on T cells, and again, across those other samples, within just the T cells, we were able to make out the cell populations that we wanted to make out. So we could see these pretty clear population of CXCL13 positive CD8 T cells. He also identified this CCL4 positive one, and we could probably go further, but this is where we left it. And I think it's not quite as good as what you can get with single cell sequencing data, but it's pretty amazing that you can do this right out of FFPE. And I'll just show you then the challenge, of course, is that all these tumors are different. Here's one where he described the tumor cells. So the tumor cells are in red here, and then there's different T cell populations in blue. And they all have very different morphologies. So I'll just flip through them. This is the second one where it's more diffuse, and there's CD8 T cells kind of all over the place. This is one where it has like these really distinct macrophage populations, which he nicely highlighted here in yellow, and where the T cells are more excluded. And then this other one where it was similarly infiltrated quite a bit with T cells, and then we also had this big river of B cells going across it, so a whole bunch of B cells in one area. So it's, to me, very exciting data because we can take those UMAPs and understand really what the cell populations are and then see exactly where they are as well. So a lot of work to be done then to figure out, okay, what are we going to say about this stuff? And one thing, obviously, that we wanted to ask was, where are the CXCL13 positive CD8s? And so he has this other method it's a way of measuring proximity. So he'll take each cell and do a random walk. So again, it's a very computational guy. Figure out how many steps does it take on average for each of these cells to get to a tumor cell, and then normalize that and put it on a log scale. And then he can sort of describe how long it takes for each of these samples to get. This is kind of one of the counterexamples. But in fact, he's shown on average that the CXCL13 positive T cells, at least for a couple of the tumors, are heavily skewed towards going into the tumor. But that's kind of an example of what we're going after. But what's interesting is that you can start to do this kind of stuff. And so I think it's still early days, but just from these 2 tumors, or these 4 tumors, and 2 that have a lot of these exhausted T cells in them, those also tend to be significantly co-localized with tumors. So, a lot more to think about in terms of analysis methods, but I think we're chipping away at this, at least with the segmentation and now with some other ways of trying to look at it. And in the innovation lab, we're doing a lot of other things. And so just another example of a cool data set, we ran this tissue microarray that has like 6 different tumor types, a couple of each. And so it's just this broad, they call it this toddler TMA, a bunch of just kind of random tumors and tonsils. And we can take all these 18 cores and put them onto one big UMAP and see all these different tumor types and be able to analyze T-cell phenotypes across all these different samples in a pretty coordinated way. So we're really excited about all the stuff that we're helping folks to get going on. And I think, again, these are using pretty standard gene panels, and I think there'll be a lot more that we can do when we also add in more thoughtful gene panels that are coming along very quickly. So then I'm going to transition to the next part here, which is, oh yeah, question, yeah, sure.
Yeah, about the mast cells.
Mast cells, yeah.
Do you see mast cell infiltration in all the tumors or only quite specific for tumors?
I don't know. Yeah, I didn't. Yeah, you come and look at it with me. I don't know. Yeah, I don't have a good answer. Yeah, lots to look at. I mean, yeah, we're generating tons of data, right? So like from these TMAs, you could imagine if you're interested in mast cells and we can delineate them, then yeah, come and look at them. Any other questions? Yeah.
For your kidney samples, what percentage was normal kidney versus tumor? Or just,
Well, we ran, so we had, so from Northwest Biotrust, we got adjacent sections. We got FFP blocks from a normal margin, and then we had the tumor. I think in the tumor, there was quite a lot of tumor there.
Yeah.
But that normal, so we had separate slides for normal and tumor. So there, I think, you can see the tumor is kind of amorphous, but on the normal, it was all normal, yeah.
Yeah, and the reason I was commenting was there seemed to be relatively few normal kidney cells, and then also,
Oh, in the tumor parts, yeah?
yeah, and also the sort of the amount that we could distinguish different types of kidney cells was maybe less than you could
In the normal part.
compartment or with that. And I'm just wondering if that's just related to the genes that were selected.
Yeah, I'm sure it's the genes, right? Again, I mean, as I mentioned earlier, because at first all they had was this breast cancer panel. And so it had a lot of genes for distinguishing different cell types there. It had a handful of immune cell genes, and then we added on what we could in terms of kidney, but we weren't thinking about normal kidney tissue as much as we were T-cell phenotypes and stuff. But this has improved a lot, right? So now they have much better gene panels that you can start with as a base, and then you can be a little smarter in how you generate your custom panels. So we're getting better at this, yeah. Any other questions about this tumor side before I go to the blood side? So I think, as I showed at the beginning, I've been really, fascinated by taking blood samples and then understanding what's happening to people's immune responses from blood. And so I've always wanted to really track down tumor-specific cells in blood. And as I mentioned also before, I showed in the tumor, even when we have tumor-infiltrating T cells and we go through all this effort, to make candidate neoantigens, it can be really difficult to find tumor-specific cells. And so out of that frustration, moving to virally driven tumors where we know the tumor is infected with the virus, and can we learn something by looking at those T cells that are specific for those viruses? And just to further highlight this issue, another way that we're trying to track this down and that what people are doing, and this is a really cool paper where if you use TCR sequencing in the tumor, and then you try to track those T cells into the blood, in fact, and we had data on this as well, and I think this was kind of an old way of thinking about this, is that if you look at in the tumor, basically you have, in this case, they did all this heroic effort similar to what we had tried to do, getting both, tumor antigen-specific cells, neoantigen-specific cells, and then other ways of identifying neoantigen-specific T cells. You can see that the tumor-specific cells end up in this particular region of the UMAP that is, dominated by CD39 and CXCL13 expression. And they called, in this case, they called these terminally exhausted phenotypes, basically. And then if you look at the virus-specific cells, like what I showed, they end up in this non-exhausted memory phenotype in the tumor. And if you try to track the clones, if you just take all the T cells that are in this sort of exhausted phenotype and then try to find them in the blood as compared to the non-exhausted memory TCR clones and try to find them in the blood, these blue lines are the frequencies of those clones that you can detect in the blood. So it's super easy to detect these non-exhausted memory phenotype T cells in the blood because they're bystanders. They're like CMV and EBV and flu and stuff. They're very abundant in the blood, whereas the tumor-specific ones, the exhausted ones, they're orders of magnitude lower frequency in the blood, and in some cases not even detectable at all. So this further highlights the challenge of trying to track tumor-specific responses in the blood. And we had similar, we have some similar data from the kidney, where again, it's this little, in this case, we had a lot of terminally exhausted phenotypes in the tumor. This is not as pretty as their example, but it's really these bystander-looking T cells that are easy to track into the blood, and they tend to have effector memory phenotype in the blood. So anyway, that's further motivation to try to look at something specific. So for the next 20 minutes, I'm going to talk about sort of this interesting comparison that we've been making between looking at EBV-specific cells in nasopharyngeal carcinoma. And then I'm going to focus on the data that were generated and just recently published in Merkel cell carcinoma, looking at these Merkel cell polyomavirus-specific cells. And the interesting comparison is that with NPC, people have done a lot of checkpoint blockade trials. And even though there's tons of T cells that infiltrate NPC tumors, you've got plenty of EBV-specific T cells. The tumors express EBV. Checkpoint blockade does not work very well. In Merkel cell carcinoma, we know that these Merkel cell polyomavirus-specific cells are very rare. The tumors are also infiltrated. And in this case, you have a very, very high response rate to checkpoint blockade, even 30% complete response rate for these advanced Merkel cell carcinoma. And that was this paper from Paul Niem's lab. And so we were actually working with the samples from that trial, the CITN 09 trial. And so we got blood samples longitudinally. And then we worked with David Cole, and we also predicted a bunch of Merkel cell polyomavirus-specific T cell epitopes. And then we ran our standard, multiplex tetramer assay on these blood samples. And the frequencies of these cells is very low. And even though we screen lots and lots of different viral epitopes, the data are pretty sparse. Nonetheless, you can see, and these frequencies are hard to read, but the red boxes are kind of where we got pretty decent hits for antigen-specific cell populations. And then the different columns are different time points for each of the patients. And you can see already just from this that we were more likely to detect virus-specific cells in the complete responders as compared to the partial responders or progressive disease patients. And that's summarized here where in the virus-positive patients that went on to be complete responders, we can detect more often these relatively rare, virus-specific T cells with tetramers as compared to the non-responders. And then what was also interesting is that if you look at the complete responders over the course of response, this is many months after the treatment, when the tumors disappear, so do the antigen-specific T cells. So that was somewhat unexpected that the memory is not really maintained in the blood, and that those cells just kind of disappear. We did have some examples of really early post-treatment where we see some blips of increased frequency, but for the most part, in the complete responders, we just see a decrease in those frequencies. And then consistent with a few other papers that have just come out, we saw that if you looked at I mean, this is just another way of plotting those data, but in thinking about it in terms of survival or progression-free survival, if you stratify by whether or not you have high frequencies of virus-specific cells by tetramer staining versus low, you can predict very well who's going to respond and who's going to have progression-free survival in this cohort. And then there's a couple other cohorts, or actually, yeah, one other cohort where this has been validated. And so because we were able to get at these cells, we wanted to look at what the cells were doing and try to see if we can learn something from their phenotype. So another typical plot for me is a UMAP plot of CD8 T cells, and then just overlaying onto this where these Merkel cell polyomavirus specific cells are. And you can see they're kind of in this region that expresses CD39. And so that was what really jumped out at us. Even in the blood, CD39 was kind of a really good marker for those Merkel cell polyomavirus specific cells. And then the other ones that we were keyed in on, and you can see a few dots in this CD103 positive population. And I'm not going to get into the NPC data too much, but in NPC, in fact, for patients that have an NPC, we see a lot of EBV-specific cells that have this same phenotype, where there's CD103 and CD39. So we're definitely keyed in on CD39 and CD103 from ours and other work in tumors, and we're kind of surprised to see them relevant in the blood. And then the other thing that jumped out is CLA expression. So CLA is this cutaneous lymphocyte antigen. So this is, David Cole also has really nicely described how this is a skin trafficking receptor. And if you look in skin, tissue resident memory T cells in skin express CLA. And so that was another unique marker. So if you summarize this across all the hits that we got for these, tiny populations of Merkel cell specific cells, and this includes, remarkably, both the large T antigen or the T antigen derived epitopes, as well as the viral structural protein epitopes. We see this really heavy enrichment for CD39 expression and CLA expression. And we had these other control epitopes to compare with, and you don't see those phenotypes for CMV, EBV, flu, or in this case, we also had HSV, which is a skin tropic virus, and so you see CLA expression on those HSV-specific cells. And again, David was the one who had previously described that and was excited about those data. And so we're keying in on these kind of phenotypes, and then looking more broadly at just the CD8 phenotypes in general. And you can see that in this one really remarkable patient where over the course of treatment and during complete response, there's this little region, and I think I'm coloring this one by CD39. There's a region of cells where there's CD39 and CD103 expression, which is nearby to where the Merkel cell polyomavirus specific cells are. In this case, they're not 103, they're CLA. But that whole little region kind of disappears during the response. So I think the cool thing is that by using the tetramers and knowing what phenotypes to go after, you can kind of zoom in on these regions, and this is kind of a remarkable example. But then we try to then take advantage of these markers to see what happens if we just use them in bulk. I don't want animation. So if you look at the frequencies of these cells, they're no different in virus positive versus negative. But then what was interesting is that the frequencies of this, so the CD39-103 outcome that we were interested in, again, mostly because of the NPC data, in fact, correlated somewhat well with tumor burden, but also percent change in tumor burden. So actually the people that had tumors that grew post-treatment, so the really non-responders, the frequencies of these cells goes up. So this was kind of unexpected that the tumor-specific cells in this case, or what we thought was enriching for tumor-specific cells, or we do think, even though they're expressing these markers that we were really keyed in on, CD39 and CD103, in fact, this population did the opposite of what I just showed for the virus-specific cells. So the complete responders had lower frequencies of these cells at baseline, which is probably related to, at least or confounded somewhat by their tumor burden. And then for the progressive disease patients had higher frequencies of these. And then in some of those, as the tumors grow, so do the frequencies of these cells. And remarkably, if we put this on one of these survival curves, you can stratify and actually predict bad outcomes. So people that have high frequencies of these CD39, CD103 positive cells predict bad outcome. And I'll just note also that there was a paper, a Steve Rosenberg paper, where they're looking at neoantigen-specific T cells in the context of lung cancer. They see this phenotype in the blood as well. So I have a few examples. And so people are keying in on this phenotype, but in fact, in Merkel cell, it's actually having kind of, even though I do think that this is probably enriched for tumor-specific cells, for this phenotype, it actually has a bad predictive value. And the opposite we saw for the CLA phenotype. For one, kind of unexpectedly, we see more of these cells only in the virus-positive Merkel cell carcinoma, not the virus-negative. So don't know why we're not having this kind of response in the virus-negative, even though they respond to checkpoint blockade well. It's very crude, but we do see some correlation between the frequencies of these cells and the frequencies of the tetramer positive, right? So we think that this is kind of a better surrogate for tetramer staining. And we did see that this combination of markers was really uniquely expressed on those virus-specific cells. And in this case, it's like the virus-specific cells as well in that they predict good outcome. So the complete responders have the highest frequencies. And then just like the virus-specific cells, this phenotype kind of disappears when the tumor disappears, and can even increase in people where the tumors are continuing to grow, and then has a good predictive value. So people with high frequencies of these cells can survive longer. And so we're really excited about this, and we have been trying to work on trying to better understand what this phenotype means. And I think, to me, it's exciting that we have a population of T cells in the blood that are indicative of response, but because of that, maybe they're involved in the response. And so what we did was sort those populations from some additional patients, and we've done bulk TCR sequencing and bulk transcriptomic profiling, and compared these different populations. And I'm running out of time anyway, but I think just the bottom line is that they both have, so both this positive, these ones that are associated with good response and the ones that are associated with bad response, they both have like an elevated T cell exhaustion signature. I mean, they are CD39 positive, so obviously they express more CD39. They express more CXCL13 tox and everything. But then the CLA positive ones are more like these kind of transitional version of an exhausted cell where they have higher levels of TCF7 and seem to be less terminally differentiated. In terms of their TCR sequences, they're both relatively oligoclonal, but we actually saw more sharing in terms of clusters of T cells for the CLA positive ones, which I think fits with the fact that they're virus specific, whereas the 103 ones, I hypothesize that they're could be specific for other antigens and maybe more diverse private antigens. So we saw a little less sharing and a little more diversity in the TCR sequences of the 103 positive cells. So that's the end of the data for this. So I just, and the nasopharyngeal carcinoma study, we're working on getting that published. Actually, Nandita Kumar is a grad student who just defended that work. It's looking really good. It's a similar thing. However, we only see this kind of CD39, CD103 phenotype as correlating with tumor burden. And I think kind of consistent with the notion that these tumors don't respond well to immunotherapy, or maybe at least EBV specific cells are taking on this phenotype that's kind of overly exhausted and not necessarily associated with good response. Because they express CD103, we hypothesize that they were actually in the tumor, so maybe they've left the tumor and recirculated, because normally CD103 is only induced by T cells in tissue in response to TGF-beta, and we think that's probably what happens to tumor-specific cells. They go into tumor, they get terminally exhausted, they express CD103, and then maybe for some reason some of them are actually recirculating back out into the blood, and that's why the frequencies correlate with tumor burden so well. But then they're also indicative of this kind of overly exhausted response that's not responsive to checkpoint blockade. Whereas in Merkel cell, we see this CD103 negative population that also expresses CLA is a little bit less exhausted and really strongly correlates with response to anti-PD-1. And so maybe these cells are coming from lymph nodes and on their way to the tumor and more indicative of something that can be rejuvenated more easily. So yeah, the overall thinking is that maybe when you're looking at tumor-specific T cells in the blood, we don't know where they're coming from. They could be coming from the tumor, and that's why they have the CD39, CD103 phenotype. And when the tumor's gone, you lose those cells. Similarly, maybe for the cells that are on their way from the lymph node to the tumor, they may be more susceptible to checkpoint blockade. And they also disappear when an antigen is gone because there's less source of antigen when the tumor is gone, perhaps. So anyway, these are things that will be interesting to try to follow up on, and we're working with Paul Neum, and they have a part of this work will go into their program project grant that they've been working on. Okay, just the last couple minutes. I know I'm late. I just wanted to mention, I always realize when I write out these slides that I have too many things going on. But we're doing more of this T-cell profiling and other tumors, this Amy. Hugh, this is somebody that hopefully people can work with. He's doing large-scale flow cytometry data analysis, and especially focusing recently on transplant, so looking at both donor and recipient in the context of stem cell transplant, some really interesting stuff. And then folks here actually know well Tony in my lab, who's been collaborating with Heather and Rebecca Gardner and Stan Riddell and Sylvain Simone in his lab to do CAR T-cell profiling. And we have some really, some new interesting data that looking at whole blood, where we think that there may be some signal for correlates of neurotoxicity and CRS. And this kind of relates to a goal that I've been talking about with Larry Fong to try to coordinate immune profiling for various trials. And so we're using this kind of an example of what we think we would like to try to do together to look more broadly across different trials in a coordinated way. And then, yeah, Ken, I didn't have a chance, again, to talk about the work with you and Nana that's being led by Ali, looking at malaria vaccine and challenge studies. And then I mentioned to some of you folks that I talked to this morning about the work that Guoyu's doing, trying to get away. So I talked about all the stuff that we're doing with tetramers, but my goal is to stop using tetramers. And so I want to try to take advantage of TCR sequencing to catalog antigen-specific T cells and then track them through their T cell receptor instead of having to stain them with antigen. And that's a collaboration with Julie McElrath and other folks, especially Andrew and Koshlin at the Hutch. So there's others, but those are the main ones that I wanted to mention because that's the kind of things that we're doing. So hopefully, in being down the street, you have a better sense of the types of stuff that we're working on. And yeah, these are the folks that did the work I was talking about. I talked mostly about some older stuff, like with Yannick. He's not on there. But Hiju and Timothy. Timothy's a medical student now who helped get the Merkel cell carcinoma stuff going. And then Nandita for the NPC work. Daniel, I talked about a lot, with the spatial transcriptomics. And yeah, the innovation lab, really the real leader of this is Anna Els. She's really great, pushing all this stuff forward. And Ozzy is another computational person that helped with the analysis that I showed. And yeah, the Merkel cell work has been really fun with Paul Neum and his whole large group of people that have amazing insight and access to Merkel cell carcinoma trials. Yeah, and I didn't talk about the Singapore connection with NPC. Yeah, and then also we worked with my old competitor, Sina Hadrup, who was on some of the tetramer stuff. So I'll stop and take your questions if there are any for a couple minutes. that it might be poor responders as well because they're better because they're even less far along. Yeah, I mean, well, I think you can't, I mean, there are, I guess what was remarkable, and you can see that on the plots, I mean, there were definitely, what was remarkable is that the frequencies of CD39 expression were so high. I mean, nothing's ever 100%, but these were quite high. But yeah, obviously they weren't 100, so there were definitely some that were negative. The challenge, I think, is, and in other data sets, if the tetramer staining is not perfectly accurate, then you'll definitely see less of a skewed phenotype. So that's always a concern, is that, you know, we have this really nice skewed phenotype, and there's some outliers. Are the outliers the cells that you're looking for, or are the outliers technical problems? But I think there definitely could be. And so that's something that we could try to quantify. I think the nice thing about these markers that are just not normally expressed in blood, so CD39 is very low typically in blood, is that gives you this sort of signal to enrich for these rare populations. And that, and combined with, you know, CD103 and CLA are also super rare. It biases, yeah, of course. But my problem is that if I don't use these markers that are so heavily skewed and rare, then if I quantify CD39 negative cells, it'll be, yeah. No. Yeah, of course. Yeah, of course. I mean, yeah, so we could try that. No, that's a good point. Yeah. Oh, hey, Heather.
Hey, Evan, that was a beautiful talk, as
Thanks.
always. So I have a question. So CD39 inhibitors have failed miserably in clinical trials.
Okay.
Have
yeah.
you looked at what they do to T cells and if they could potentially promote? Because you see a loss of this in patients that respond to checkpoint inhibitors, and an increase in patients that do not respond. Is that correct?
Well, so the frequencies of cells that have 39, I think, is indicative of the tumor-specific response, we think. I mean, it is definitely associated with exhaustion. I mean, if they co-express 103, then that's the bad predictor. But yeah, I think David's question about 39 negative cells, we haven't looked as much into. But I think in this case, I do think that, you know, it's a marker of the good cells as well as the bad cells.
Okay.
But yeah, in terms of what CD39 is doing, I know this is something we always thought about, you know, when it was shown as such a good marker. I think the problem is that CD39 is all over the place, right? It's expressed by almost all B cells. It's expressed by tumor cells. In the tumor, it's all over the place. So these inhibitors are going to inhibit all kinds of stuff. I think its role on the T cells is kind of a negative regulator there as well. It's some kind of feedback mechanism. I don't know how important it is. So maybe it's not, you know, I think in our case, it's more of just a marker of terminal exhaustion. And like other markers of terminal exhaustion, it's kind of a negative feedback. It's one of many ways that there's negative feedback. And so maybe that's why, you know, blocking it is not itself so important. I think probably, and more of the motivation for blocking it came from, you know, its abundant expression on tumors and how it can, you know, promote degradation of ATP and have adenosine, which is immunomodulatory. But yeah, how T cells, what role T cells play in that whole thing, I don't know. Yeah. I think most of the time they're a negative sign. I mean, they associate with colder tumors. Yeah. They also express really high CD39 as well. So I think that's part of the way that they're regulating the environment. But yeah, I think for the most part, they're a bad associate in the tumor.
So Tregs can be CD39 positive?
They're almost always, yeah. Mm-hmm. And like in blood, you see it. Oh, you could pull them out, take them.
Identify the Tregs and CD39, would they be associated with tumor progression?
In the tumor, I think they would. In the blood, I don't know. Yeah. I think they would be, yeah. I mean, I think, yeah. And I mean, I think another funny, you know, in the CD4 case, you know, with CD4s, when they become I think there's this tricky thing in that tumor-specific CD4s can turn into what look like Tregs. They start to express CD39, they also express FOXP3. And so there's that tricky thing, is that not all FOXP3 T cells in tumors are necessarily even Tregs. They're just kind of these chronically activated CD4s that express more and more of these inhibitory markers, including FOXP3, and CD39. Okay, well, thanks.
Thanks a lot. I think we're over time. That's awesome.